Hi, everyone, and welcome to our IndieBio event about data and biotech. I'm pleased to be joined by three amazing founders who are all using data in very unique ways today. Joining us today are Amal Grafstra of Biotech. Amal, hi, thank you so much for having this conversation with us.
Sure.
Also joining us is the CEO of Annika Biosciences, which some of you may know formerly as Carver. It's probably the founder here that I've known the longest since we met back in 2017, although I wasn't able to be part of his journey through Rebel Bio. This is Vishal Bhuyan. Hi, Vishal. I'm sorry, Vishal, if you were saying hi, I didn't hear you.
Just yes, hi, yeah. Sorry.
Yeah, no problem. Just want to make sure you're not on mute for that conversation. Great. And our third founder is Adam Kirkowitz of Biomage, a company turning every research scientist into a bioinformatician. And I'm also pleased to say was the 1,000th investment from SOSV, the parent fund behind IndieBio. So Adam, hi, thanks for joining us.
Hi, Julie. Really good to be here, and so glad that we're part of SOSV's journey.
Yes, well, it's only just beginning since you were part of the most recent IndieBio New York cohort. We're really psyched to have you here today. Great.
Absolutely.
So I haven't introduced myself. I'm Julie. I'm the communications director here at IndieBio in New York. IndieBio, for those of you who are not aware, is the world's leading biotech accelerator. We work with companies that are using biotechnology to solve the world's largest problems in human and planetary health, spanning every single thing that you can think of that touches biotechnology. And during the 4-month mentor intensive program, companies receive mentorship, they receive workshops, they receive $250,000 in capital to move their tech forward. And as you may see behind me, lab space, we've recently moved into our New York lab, and we're very happy to be here. Today, we are going to focus on the many ways that biotechnology can touch on data. And I want to point out a couple of different things about the website that you're viewing us with in case you haven't joined any SOSV or IndieBio events recently. We have this lovely platform where if you scroll down, you'll find information about the companies, the founders, the websites of these different companies if you're interested in exploring more about those. And importantly, we want to make this interactive. I know a number of you put questions into your registration, and we've certainly incorporated those into today's event. But if any additional questions pop up, there is an interactive chat box. By all means, add questions into here, say hi to one another. We know there's a number of registrations from all around the world. So if you want to say hi from whatever country that you are visiting from, if you want to add your LinkedIn information to connect with like-minded folks, just go ahead and make this as interactive as possible. All right, let's dive in. I think that the best way to begin the conversation today is for each of our founders to describe what it is that your company makes as its product, how data is incorporated into this product, either as the final thing or as part of the value proposition. And if you could maybe touch on your business model, that would be, I think, an additional valuable piece of information. So I'm going to hand the mic over to Amal to kick things off. Amal, if you wouldn't mind, just briefly in a couple of minutes describing Biotech.
Yeah, sure, no problem. Biotech is a company that's focused on secure, cryptographically secured, implantable transponders, so RFID and NFC transponders. And we make, primarily at this point, we make transponders for the purpose of secure identity applications. So the data involved in doing something like that is mostly about shuttling cryptograms between platforms and services and APIs to the chip through devices like smartphones with NFC, for example. So there is oddly a lot of concern about oh, it's a microchip implant. Are you going to track me? Who's going to steal my data? Those kind of questions. And the reality is those questions are based on kind of miseducation that comes from Hollywood movies. There is no tracking capability. We don't track users around. In fact, the phones that you use are much worse at that than anything that you implant, at least today, could ever be. So really our focus on data is really connecting systems and making people more privacy aware, more private, and more secure. In the future, and in fact, I have a prototype here that I just put in yesterday. We are expanding into bioinformatics, that kind of thing. So medical data collection, that type of thing. And that's where the question really will become, what kind of information collected, how is it secured, how is it shared, who's it shared with? And in that, you look at companies like Fitbit, where they had non-medical status, and it was just a kinesiology device, and you could share the data with anybody, and the company was using that data to make assessments of their customers. So as they transitioned into a medical device company, there were some laws that kicked in, and they had to change the way they handled the data. But these are all questions that we are now grappling with, and particularly with GDPR and like all the EU personal data protections that come along with it, and including US HIPAA compliance, that kind of stuff. So it's a very interesting time for sure.
So that's very interesting. Thank you so much for kicking things off. I was going to ask a question, but I think you may have answered it about where these devices will be implanted. But maybe you could talk about how many one might use and whether you could use different frequencies in order to carry different types of information.
So initially I started out just selling basic transponders through a company called Dangerous Things. So these are for kind of like the early adopter biohacker types. But they're not secure. They're not really able to be used for larger applications like banking and this kind of stuff. So, RFID in general as a technology is kind of an umbrella, and under that umbrella, there's a lot of frequencies and data protocols and standards. So, right now I have 6 devices. You can't really see them or tell that I have them, but I have 6 devices. 7 now because of yesterday. But the point is that when it comes to compatibility with existing systems, badges, and things like this, when I was at IndieBio, I actually brought some of these transponders and we implanted some of the cohort so that we could put our badges on our hands and then go through the building with our implants. So those kind of applications, required a specific kind of chip with a specific compatibility with the existing system. The goal with VivoKey, of course, is to kind of become more of the Apple of the implant kind of concept, where we will release a door lock, we'll release software and services that all work with VivoKey implants, and it just works, right? Rather than having to worry about having 6 or 7 implants, we want you to have a VivoKey implant and then use those services and devices that it works with, including things like applications where we might partner with practice management companies, where you walk in, you check in just by kind of putting your hand into like a little ring, and it collects information about your basic setup, blood pressure. Well, maybe not blood pressure, but just some things that we can share, but also securely cryptographically checks you in. It opens your exact medical record, not like some people might have 2 or 3 medical records in a system because one day they, we can't find her, they spelled their name slightly wrong or something, and they open a new record. So all that goes away. You're cryptographically, unequivocally identified correctly, all with just like putting your hand in a thing and you're done. You go in the doctor's office room and you do the same, and your record comes up. There's no question. This is your medical record, that kind of thing.
Very, very interesting. And I see that there's a lot of different medical applications, but I was just thinking how convenient it would be to have one of those things, as we've had to use these badges at just about every entrance that we go into nowadays.
Well, talk to me after. We can get it set up.
Awesome. All right. Next, can we hear a little bit about how Annika is changing the security of the supply chains as the world becomes more and more global?
Sure.
Michelle?
So sort of on a high level, what we do is we use microbes as tracking devices. So what we do is we convert data, digital data, into strands of DNA. We insert that little bit of DNA into a microorganism, a probiotic microorganism, to be honest. And then we can sort of apply that organism and sort of have it hitch a ride on any food or agricultural product or really anything through the supply chain. And the reason we use a microbe to do it is because we engineer it to go into a spore. So a dormant state that allows it to be impervious to high temperatures and UV light, sort of protect that DNA barcode through transit. And so why this is important is because you can spray romaine lettuce, for example, and you can mix it around and wash it and treat it, microwave it. You can have it decay for a month, and we can still re-identify sort of each leaf back to its origin. And so based off the different kind of requirements in different supply chains, that microbial tag allows us to get just really granular information throughout the entire transit, without having to worry about things like, cross-contamination or of tags, or rubbing off from one thing to another. So it's just an incredibly hardy package of information. And then we couple that with a quick readout device that allows you to not have to send things out of the supply chain to a lab, which is how they're done now, but just sort of verify these types of quality assurance and quality checks on the spot.
So I'm guessing that using these spores makes the organism metabolically inert, so you don't have to worry about things like the low level of DNA mutation as organisms
they're
may.
totally inactivated. So we've removed, I think, 3 or 4 genes responsible for that germination. So really what we have is just a particle, in essence. It's just inactive.
And you mentioned that it's a probiotic, and you used lettuce as your example. So is this something that you imagine is going to be mostly used in food supply chains? I could see that being extraordinarily useful, but perhaps you can even branch out into non-edible things.
Yeah, so we've worked with De Beers on diamonds. We've gotten inquiries about tagging explosives. To be honest, picture a big mining company whose explosives end up in a civil war. We've heard everything. Both organic and non-organic in terms of applications. We're just focusing this year particularly on romaine lettuce because of customer demand, but the applications are actually just really endless in terms of how you could apply this to a number of different things.
Absolutely. Wonderful summary, and we'll get back with additional questions, but I want to move on to Adam. Adam, can you talk a little bit about Biomage and the software platform that you've developed and are now onboarding customers right and left?
Absolutely. So at Biomage, what we're building software which can turn every biologist into a bioinformatician. And the reason it's important is because there are a lot of very bright people at a lot of different universities, including one of our customers, Harvard Medical School, who can use biology to discover drugs for diseases such as cancer, cardiovascular disease, or diseases like Alzheimer's. However, the type of technologies that they get to work with nowadays actually produce a lot of data. So biology used to be historically a low throughput science, a science where one experiment would correspond to one data point, usually a smudge on a piece of gel. But nowadays we live in a world where a single scientific experiment can produce even 200 million data points, and it's really just this journey from a low throughput science to a high throughput science, which has made it really difficult for a lot of biologists, even in those top tier institutions like Harvard Medical School, to just really deal with the volume of data and really to be able to analyze it. And at Biomage, we've built an open source product that makes it easy to analyze a specific type of data called single cell RNA-seq data. And the data that we analyze doesn't actually. So one of your questions here, Julie, at the beginning of this round of questions was like, how do we incorporate data in our product? Actually, we don't create our own data. So we're not a data company like Google is a data company or like a Facebook is a data company. We enable people to understand their data. We enable research scientists to understand their data, through which they can create new discoveries that can lead to new treatments and human disease. And the business model question, which was also something you asked us at the very beginning, is I think that's actually really interesting, because for an open source company, Somebody could tell us, you're an open source company, how can you make any money? To which I like to answer that the fact that you own a horse doesn't mean that you don't have to pay for the for all the things that are connected to you owning the horse, such as the feed and other elements. So for a company like Biomage, what we sell to our customers is we sell additional feature development, we sell deployments, we sell all the things around the software, which allows us to have an open source bit of software that people trust because it's easily verifiable. So that's really the business model.
Very, very interesting. And thank you for addressing all three of my questions there. I know I tend to ask more than one question. So it's more on me than on anyone who doesn't answer all of them. But I love this collection of startups because I think that you guys are all using data in extremely interesting, applicable, but varied ways, right? Like I think many people, when they think data and biotech, think more along the lines of the Google model, right? Like collecting a lot of things and being able to find them. But we have people who are working on giving people ownership, I guess you could say, of their data with the RFID chips and other types of implantables, being able to secure the location or the source of various places in a world where we're increasingly globalized, and helping people to sort through their own data and giving them ownership in that sense of their own analysis. So really, really interesting collection of people working with data. Now, there is one question already in the chat, and it's addressed to Amal, but I think that this is one that all three of you could answer. The question is, Amal, what is the need for these devices to be implanted? And I actually want to reword that a little bit in terms of how did you, Adam, Vishal, Amal, how did you see an opportunity to build a business, right? How did you know that this was an area that you could create a product that people would pay money for? And how much iteration was there around building that product?
Sure.
But since the question was first to you, Amal, I will pass the mic back over to you.
Okay, I'll try to be quick, but essentially the short story is at around 2005, I was working in a medical office doing IT support for small clinics, and I had a door that would just lock behind me. It was an emergency door, and it wasn't meant for egress, but I used it that way. But when I'm working IT after hours, 2 in the morning, and I pop outside to get something from the car and forgot my keys, now I'm stuck, right? And I can't, I can't do anything. So I just over time got really irritated with the idea that, keys are this archaic form of, cut metal identity token from like 700 BC that represents me to the door. And I wanted the door to just know that it was me and let me in. So the first application was access control. It was just to be able to go through the door without much hassle and without, more importantly, without having to carry or manage anything. And so, to address the first part of the question, why implant these things versus carrying things around, and the real answer is management. You might lose your keys once a month or once a year or never, but you always manage them. You always have to manage your wallet full of tokens, which are the payment cards, the identity cards. You have to manage your key ring, which is full of cut metal tokens that represent you to locks. You have to manage passwords, which are identity tokens, which represent you to systems. And so the idea that you can simply and cheaply be able to kind of replace all of that stuff with something that is managementless, frictionless, it just goes in, it's safer than an ear piercing to get, it's quicker than an ear piercing. It just made sense to me. So, I put this thing here in my hand. This is since 2005 and I still use it today.
That's a long life. I just wanted to comment on that. That's 15 years of one device. Most devices, when we think about what we purchase, 2 years, maybe 4 if you're lucky, right?
Yeah. I mean, we design our stuff to at least go for 30 years, if not more. VivoKey stuff is good for 50 plus years. So, but the point being that, I did it just as a convenience application for myself, but then other people got wind of it and got very interested. And then, as the maker revolution started happening and people, getting into Arduino and homebrew electronics projects, I really started to get a lot of inquiries. And I was like, man, I got to figure out either I got to ignore this or I got to, wrap a business model around it. And so I started Dangerous Things for early, biohacker types in 2013. But even rapidly by 2018, which is when we participated in IndieBio, the type of customer changed dramatically from like, give me the data sheet to what kind of cool things can I do with this, right? And they weren't familiar with the technology, but they were very interested in doing it. So, VivoKey was launched to solve not just where are my keys problem, but also bigger problems like international identity, secure banking, like all this kind of stuff. And really the concept, again, when you kind of talk about, well, I don't mind putting my password in, I don't mind, doing this stuff. I noticed that all the panel members have glasses, right? Well, there's people that don't want to manage those glasses. They get LASIK, right? It's changing their body to fix a problem, or in their case, in some people's cases, you can augment yourself. So in our case, we're augmenting your human capabilities with a very simple chip implant that does its job. I like to say, you're sitting there now and your kidneys are working hard for you, but you don't give them a second thought. This is exactly how this technology works. It just sits there, it's accessible, always there, you never think about it, it's frictionless. For me, if I can get rid of, and for 16 years, I've never had to manage my keys. That's 1/3 of my phone, wallet, keys, typical management burden. If I can get rid of 2/3, get rid of the wallet as well, get rid of passport, like there's all these things that I could do to kind of enhance my personal freedom without compromising convenience and increasing security at the same time. Why wouldn't I do that? So I hope that answers the question.
It's great. It sounds like instead of having to go out and do customer discovery, customers were actually coming to you and saying, can you do this? Which is, I think, something that every founder would like to happen.
Yeah.
As you can think about going into some of these medical spaces, are there different types of compatibilities that you have to consider or regulatory paths to work with the FDA to carry this information?
Yes, somewhat. So the idea of FDA medical device, categorization is interesting. So you can make a thing, right? So I mentioned Fitbit earlier, but in our case, we can make a chip that we can use with third-party systems, our own systems, NFC phones, all of that stuff, and it doesn't qualify as a medical device because it doesn't tick any of the 3 requirements or conditions under which a device becomes a medical device. And the biggest one is it is not used in diagnosis or treatment of disease. Because it's not used in that fashion, it's not required, just like body jewelry is not required to be FDA approved. But going into the patient identity medical records application, the use of this technology, that dictates we need to go through FDA approval, which in this case is not all that arduous. There's already a pre-existing product that attempted to enter the space, and so we can, they had a lot of problems with how they approached the market, but we can attempt to do this, with a basic 510K filing, which is saying it's exactly this or extremely similar, right? And so they already had approval in 2004, actually. So it's not going to be an arduous task to do. It just matters that we have to, kind of space those regulatory hurdles out and overcome them when applications are ready.
Yeah, that makes sense. Excellent. Thank you so much. Vishal, I know that you have been working with different supply chains, as you mentioned. I wonder how you saw that there was a need for better management of the sourcing and the following the cargo as it's moving from one country or from one container ship to another, and how you thought to use microbes and DNA as the answer to that question or to that problem.
It's kind of funny because you're actually part of this story, but I was actually working in finance. My background is in finance and I'm not a scientist. I was making angel investments and just doing side projects. One of those things was a CPG product, a consumer product, a snack food made out of lotus seeds from India. And they were actually sold all over New York City. There were like Google headquarters and Facebook, just like a fun pet project. And we were importing like, thousands of pounds of these things from India. And they kept getting held up at the border. Particularly, we had to, in one instance, discard 3,000 pounds of them and work with the FDA because of a pathogen outbreak and a banned substance that was sprayed on them. And so this hobby became a complete nightmare, where I was spending thousands of dollars on something that's just gone wrong. And so working with the FDA to do all these assays to determine sort of what happened to this product and actually what its true origin was blew my mind. Like, why don't we know this? This seems like common sense stuff. And this problem just kind of like seeing the scale of this in the dock, at the shipping, on the cargo ships was amazing. Because when I was talking to the FDA, it's like, yeah, not everything gets checked. This problem is way bigger than just this container. It's everything. And sort of the complexity of the food system just became very clear to me. And the fact that you've got to do like dozens of different tests to verify things, both that touch food safety and traceability, just seemed like a massive problem. And sort of at the same time, I'd taken a class with Julie at GenSpace called Biohacking 101 or something. And the two things just sort of merged in my head, which was, what if there was like a friendly microbe that we could use that said, hey, this is the product that you think it is. And that was sort of really the synthesis of the idea, which is, I saw all the different ways people were using sort of synthetic biology. And I thought if we could leverage this as a utility, as opposed to something that's going to necessarily do something, whether that's, add nutrient value or any of that, if we just literally use it as a utility, this could solve a problem that I've kind of come up close and personal with on this really big scale. So that was really how it started.
That's very interesting. You were kind of your own customer with a problem, right? And then
That's
you
exactly
recognize
right.
that's very interesting. So similar question that I posed to Amal, as you developed your product and started to deploy it, were there certain regulatory hurdles? I'm thinking more along the lines of EPA than FDA here. If you're going to take, even if it's an inert microbe, are there certain hurdles that you have to cross in order to use that product?
So the biggest one for us has been FDA, and it's really just obtaining GRAS designation. But because these are inert and because these are in a very faint kind of LOD, so the amount that's being sprayed on is very minor. That's really the things that we need to prove to the FDA. So but the GRAS certification that we're getting, the no objections letter, we're very confident we'll receive that pretty much by the end of this year. Again, because they're inert, the edits that we make are very small. We're talking about, about 50 to 100 nucleotides. The fact that it doesn't germinate is huge. So that's all reasons why we're very confident in that. In terms of EPA, it really depends on the fact that they're not germinating is the biggest reason we can circumvent that, and then where it's being applied in the processing or in the supply chain. So in the lettuce, for example, it's actually post-harvest and it's mixed with the wash water. So it's actually not touching in the field.
I see.
There's one reason the EPA is not involved. And that's basically the case. A lot of it doesn't really need to be at the field, but just post-harvest or post, whatever that system is, post-mining or whatever it is. So that's usually how it's added in.
And so you mentioned very low dosing. Like, can you tell us exactly like how many? I mean, they're not even really microbes. They're like microbial parts, almost like a shield for the DNA, right? Because you have to protect that DNA, right?
The best way to think about it, so it differs between supply chains, but if you take lettuce, for example, a couple of drops of the tag in, let's say, 2 or 3 liters of water would cover 200 pounds of lettuce, for example. So really, we're talking about very faint amounts of the microbe or the microbial tag.
Okay, and a question for you from the audience: Can you use these to verify that these products have been ethically sourced? I imagine it depends on who your customer is, but
we've actually, so in cacao and coffee, this is a big issue, especially in Europe and the United States, where like ethical sourcing is legislated pretty much. So we've done things like this was one of the coolest demos, which was we took coffee beans, roasted them, like brewed cups of coffee, and then like traced them back to the origin to show that these were ethically sourced cups of coffee and these weren't. So we like in coffee in particular, I think there's like 22 different certifications that they need, and you can embed those, so to speak, in these tags. The authenticity. The other interesting thing I discovered is there's a big black market for these certifications, whether it's ethically sourced or whatever, where they trade actually, so you can slap them on your unethically sourced coffee and still push that off to the big buyers and the big importers. So the ability to tag the actual product and not the packaging is just incredibly helpful.
Okay, well, that's very interesting. And kind of gets into the next area I want to dive into. But first, I want to make sure that we talk a little bit about the origin story of Biomage and how, Adam, you were able to identify a need for the product, this software platform, as well as some of the bells and whistles that people are willing to pay for as add-ons.
Sure. Let me address this in a second, but I wanted to ask Vishal a question actually about his tags. When you did the coffee experiment, did you tag the beans before you roasted them?
Before we roasted.
Oh wow, that's amazing.
What's amazing is that the spores are just in the spores that we use are just, they cling to organic matter really well, I mean, especially grasses. That's why they're really good in things like leafy greens. And that's just a property of this. And so one of the things that actually, to go back to the earlier question, Julie, was I remember sitting in that class and someone started talking about spores, or at GenSpace, someone was complaining about spores actually, and about how hardy they were and how like they're such a pain to get rid of. And I was like, huh, that's interesting. Cause I was like, that would be the perfect way to protect any sort of tag.
Mm-hmm. How can you flip
and
that?
So I was just like, oh, how interesting.
Oh, well, yeah, absolutely.
And looking a bit more at your technology, Vishal, do you PCR, do you amplify the tag bits of the spore before identification?
We do, but we don't use PCR. We use a different type of amplification that I don't know if I can mention because Ellen will probably kill me, but we use a different type of amplification. So we don't need to do it using a PCR or qPCR.
This is being recorded, Vishal. So.
I can appreciate the careful nature of your word choice there, Vishal.
Now for jumping in, I also had a question. Just quickly, have you sent spores through the digestive systems of any animals?
That is a great question. And yeah, that's a really good, it's a big area of interest that we're currently working on. And again, I don't know if I can say more about it, but there's a huge need for that. Because if you think about linking, just in a contamination, you go in, you're sick, linking the sample, the human sample to the actual outbreak and the location would basically truncate the time of a recall investigation from like 3 months to like an hour.
Yep.
So,
cool.
Yeah. So ongoing, stay tuned. Is that the
message
there, Vishal?
It's like so raw that I don't know what I can say. But
no problem. I can see you looking over. I don't know if Ellen's there. Okay, then no worries.
So Julie, I haven't been trying out to weasel out of answering.
Yeah, I know. I love how engaged you guys are. Jump in with questions. That's fine.
I'm just genuinely interested in this technology. So back to your question. If I recall, this has been really just about our journey in terms of realizing that there was a customer need for what we were building, and I think this is a great question, and so many times, a lot of entrepreneurs I know, a lot of early stage founders I know will spend all their time trying to build a solution for a problem which doesn't exist. So that's a super important question that, for us, the answer really came from a few consulting projects that we did to verify the market need before we built the software. And the consulting projects would create exactly the same type of value to the customer instead of just a couple of hours of interacting with the software. This would be about a week of us running an automated pipeline that we've built sort of for our own internal use. So we were the software as consultants, and that's how we validated the need for the analytical services that we provided. And our premise here really was that if customers are willing to pay $5,000 for this consulting service that we're offering them, they will likely be willing to pay a similar amount of money for the same amount of value that they're going to extract from the bit of software that we're going to build. And this, so far, from kind of early closed betas that we did without software, this assumption seems to have been proven correct. But we are really waiting for our open beta, which will be at the end of April, as we finalize our software to really learn whether this assumption was true. which is so we still are waiting on verifying this market needs. The one thing I'm going to say is, obviously, out of the three businesses here, Biomage is the youngest, because we only started last year, in April last year. So we're still relatively early on in our entrepreneurial journey.
Excellent. In the people who you've interviewed, let's say the scientists who are using the software at the institutions that you mentioned, do you find that there's particular need within different, let's say, subfields? You mentioned developmental biology. I could see this in neuroscience or other types of specific areas of research? Or do you think this is a broad, everyone in biomedical research would have some sort of use?
So it's a great question. So the way I would say is, there's about 50 to 60% of the analytical needs when it comes to specifically single cell RNA-seq data analysis. There's about 50 to 60% analytical needs which are shared across different customers. There is going to be always this around 40% of needs which are really customized. And for these customers, we are working on a system of plugins that they can build on top of our software solution, that their bioinformatician can build on top of our software solution, hopefully provided back into the open source community so that others can use them as well. And this is really what's working out for our product. I suppose a kind of interesting way to approach this, and I appreciate this is going to be very different from how a lot of other businesses will be built. What we're doing is we're using our kind of base of researchers as a sort of the best proof of the usefulness of our product. And the kind of business model that's often mentioned here is the flywheel sales model, where the people that you initially target with your solution, with your software, will act as sort of early adopters, and will take your software to places. Will act effectively as a sort of free marketing. Will take your software to places where you'll be able to derive bigger revenues, so sort of more business to business customers, like pharmaceutical companies, like the clinic, like large government sponsored biobanks. So we're really starting small with researchers, and we're hoping to use their reputation and use their influence to get into those more exciting, bigger business to business opportunities.
Yeah, excellent. Kind of grassroots, like seeding the scientists of tomorrow who will go and work with those organizations.
We call it the Trojan horse approach.
All right, so now I'm going to open the floor rather than ask a specific question. So we'll see, it's a little bit of an experiment. I know in Zoom times, we're all a little worried about stepping on each other's words. But one of the most common questions that was submitted with registration was, how do you demonstrate as an early stage company using data that you are an ethical company? So getting a little bit into what Vishal was talking about, how do you show people, or I guess, how do you garner their trust with their data, or with whatever data that they are procuring through your services? And there are so many either big strategic visions or maybe even tactical means that you may be using in your own companies. I welcome anyone to kind of just kick things off with this.
I can go first. So we actually we don't know what the data is. Like, that's how we kind of pitch it to the customer, which is, this could be a barcode, this could be your mother's maiden name. It doesn't really matter to us. You just tell us what you want in terms of, like, basically length and how many. But we don't know exactly what you're we know what you're tagging. We don't know necessarily what you're tagging it with. So whether that's your own internal serial code or whatever, that's one way we can just say, hey, look, you want to tag all of these lots of lettuce or whatever it is. but we don't know what that actually represents.
Do you mean, I'm sorry, Vishal, just to make sure I understand, do you mean that you at Annika don't know what that sequence is that you're
We
tagging?
know what the sequence is, but we don't necessarily what that sequence is representing, right? So they could
I
say
see.
07582. Like we don't know what that necessarily is.
Right.
We're just
Okay.
sticking it into a sequence and we're sticking it into the organism, but that could mean a host of different things to that actual customer. And so while, you know, if it's a lot number or batch number, it doesn't really matter. There's no security issue as such, but if there was something sensitive, we still wouldn't know. So that's how we kind of address it with them, which is you're just telling us how many you need. We create the unique sequences, the unique strains, so to speak, and then we assign those strains to whatever unique situation that they have, if that makes any sense.
Vishal,
Yeah, I see.
Vishal, do you let your customers choose the sequence that that they want? Or
No,
would you-
so we're just creating our own sequences and then basically just linking them essentially to whatever the digital data that they want is, or whatever the serial number that they're looking for.
like one would imagine that perhaps more important than a sort of specific message that's encoded in the DNA will be uniqueness of that message, so that other companies or other users of your product do not come dangerously close to
Exactly. We have almost an infinite coding space, as we call it. So we use both sequence and sort of positional. So we use actually multiple ways to create the coding space. So we have very distinct differences from one barcode to the next.
Fair enough. That makes sense.
Adam, do you have any customers who are working with, let's say, patient data who are worried about, making sure that the information that they have is not going to be misused in any way?
Yeah, no, it's a great question, and it's something that keeps coming up regularly when it comes to genetical data or genetical information of humans. As we stand, the industry standard, the academic standard, is that transcriptomics data, for example, that's linked to a research paper will be made publicly available to any researchers, to any bona fide researchers in the field after the paper is published, which is a very, very valuable. This is very valuable for science because data likes to be open, and with open data we can achieve more quicker. And the things that when I say we can achieve more quicker, I mean things like creating new treatments for diseases, diseases like cancer, cardiovascular disease, diseases like Alzheimer's. So these are some huge challenges that as humanity we have to deal with, and the way we deal with those challenges is through academic research, through academic researchers creating new drug targets, creating other sorry about that, creating other types of understanding, of human understanding that can really help us.
Adam, that's funny because I almost felt like because I was hemming and hawing even as I asked the question, because I was thinking the answer that you were going to give would be, well, we don't deal with the data, it's other people's data, and we are just a tool that overlays the data that, of course, is going to be held securely in whatever database it is. I don't know if that is a true statement or not, or whether does Biomage actually interface with some of this data?
So no, very much so. These are the researchers' data, but we still are a part of this trend. We're still a part of this whole debate, and we I very often find myself having to take a particular view, and in defense of those open source repositories with data that researchers submit, potentially patient information to, there is in fact. We live in fact in a world where actually none of us really has all that much control over our own genetic information, because in a world where so many people have actually used services like 23andMe, which law enforcement can actually get access to, there will always be somebody who's even related to us who submitted their data, and as a result, we can be found through that connection. So it can be a second cousin or a distant cousin or a relative. So there's definitely it's an interesting debate where privacy concerns are pitched against scientific progress and against human health, and it's sort of like how we position ourselves and decisions that we take with response in relation to how we handle that data. That's actually really, really important.
It is, and I'm glad that we're asking these questions in panels like this, because of course every piece of technology is a double-edged sword, and it's how it's used that really defines who we are as human beings. And now I want to turn to Amal, because I have a question more on the side of access in terms of whether people would have access to this technology. It seems like you might need a professional to even put those types of biomodifications, I guess we'll call them, into your body to make sure that it's done sterilely, whether that's cost prohibitive, whether that's something that people could have done against their will under worst case scenarios. What are your thoughts around that?
Actually, Julie, can I add one thing to that question?
Yeah.
Also, how do you take it out?
Sure.
Yeah.
Well, I was kind of, I'm kind of amazed at how similar our companies are, Vishal and mine, in terms of what we're doing. We're identifying things, we're using tags, RFID tags, biotags, there's a lot of similarities there. And in a lot of ways, we kind of handle the data similarly. But, the reality is, yes, you need to go to either medical professional, like a doctor. Your family doctor could do it, but we also partner with body piercers, because body piercers are everywhere. They are trained in aseptic procedure. They work out of licensed and certified studios, and they're cheap, right? They're $50 gets you a piercing or a chip. I mean, it's very straightforward. So in terms of, again, like safety and health concerns, an ear piercing is actually riskier than getting a chip implant, because the chip implant just lays right in the fascia tissue between dermal and muscle layer. You just lift the skin, it's a 5-second thing, you're over and done. And then within 20 minutes, your Band-Aid can even come off. You can wash your hands and take a shower, it's fine. Ear piercing, right? You put a hole through and you put metal, and then you might be allergic to that metal, and then your body has to heal around this metal, and there's weeks of time where you have to be very careful and not get infected, and you could end up with a keloid scar or something like that. So in comparison, it's very low risk and also low cost, to have it put in. Taking it out is similar. You would just essentially, I've had several things in and out because I experiment with prototypes and things, but it's very simple. So it's a cylindrical tube, right? 2 millimeters by 12 millimeters typically. And you would just essentially go to the doc or any random GP, and you just press, when you press down, you can kind of see it pop up there, right? Right? Mm-hmm. So they would just make a small incision, you press it out, and it just comes out like that. Now, when it comes to like animal chips, like pets for dogs and cats, they have a biobond or paraline, some kind of porous coating around that cylinder, and the tissue grows into that coating. It locks the chip in place. And for animals, that's necessary because their scruff, the fascia tissue, is very, very loose. You can pick it up, and it's like a lot of leeway there. So they need that kind of locking in. Otherwise, the chip would just migrate around, and you would not know where to scan them. But for humans, our fascia tissue is very dense. We don't need that. But the upside is replacement or removal is very, very easy. What was the other part of the question?
Access. I was just wondering, because what about people who want access and can't get it, or?
Oh no, I remember. Yeah. It was the against your will kind of thing. So
yes, yeah.
So really, that's not. sadly, we are, I've been contacted almost regularly for 16 plus years by people who are clearly developing or in the early stage of developing schizophrenia. They're hearing audio hallucinations, they're hearing their own thoughts, they're being monitored, and people are harassing them because their own brain, they're hearing their own brain, but they don't know that it's their own brain, and the voices that are being made up. It's like dreaming, but you can't escape the dream, right? So they go online, they search, and they're like, people are getting chipped against their will, and then mind control is happening and all this. And I mean, hundreds and hundreds and hundreds of people I've talked to, and the sad result is that by the time they get to me, because they've found this chip implant is the nefarious thing, they're unhelpable. Like I've reviewed MRIs, X-rays, I've spoken to people on the phone, Zoom calls, like nobody, like they're already too far down that rabbit hole to escape. And so they're convinced they have a chip. They're convinced that it's doing mind control of some kind. And they're just no talking them out of it at all. And so the idea, though, that you have to ask yourself, like, if you're chipped against your will, for one, the technology doesn't track anything. The technology isn't GPS. It's, in fact, completely inert when not encased in the small magnetic field generated by the reader. There's no power. There's no battery. I can't do anything. There's definitely no neural interfaces, and we're not Neuralink here, you know. So all the things that they say that these people say that a chip implant could do, and would be reason for maybe implanting someone against their will, is not possible. It's only possible in Hollywood movies, right? And so, you know, we want to get to that future. We want to be able to cure paralysis and cerebral palsy and all these things you could do if these devices could do that, do what they're saying, but they can't. So there's no reason at all to chip anybody against their will. It'd be pointless. You would be giving them a gift. I mean, you know what I mean? There's no benefit for the perpetrator, right?
And in fact, you said that people will come to you with their, let's say, MRI or CAT scans. The chip would not even be able to interact with those.
They'll point at something that's a little wisp of something, and I'm like, no, that's not a chip. I have a very famous X-ray that's out there that shows very clearly what the chips look like. It's very easy.
The time, by the way, to like talk to people.
Yeah, I mean,
I find that very.
Even yesterday, it was a few days ago, somebody had contacted me, and they were, you know, it's usually a big long email, and I kind of skimmed through them, and they seemed worth responding to, you know. So I said yes. Like they claimed that they had a professor that they were talking to and a neurologist. So I'm like, great, this is great. You have professionals. I'll talk to the professionals. You link them to me. They responded, thank you so much. I'm so near suicide. I just can't handle this anymore. And I was just like, just get your neurologist to talk to me. We'll try to sort it out. But the moment I tell them it's not a chip for all these reasons, you're not the king of Brunei, you're not Elon Musk, there's no reason to, even if this technology existed in like a secret lab, why put it into you? Who are you? What value would there be for that just to harass you with voices? It doesn't make any sense on any logical level, but the moment that I start diving into that, they retract, and they're like, no, it's a chip you can't see on X-ray. The doctors are in on it. And it just kind of it devolves from there. But I still try to make an effort, at least, to try to get these people directed toward mental health professionals. But what I'm also finding that it's difficult because a doctor can't, I mean, it's from the 50s, right? Where you would put somebody into an institution, but you can't do that anymore. So the doctor really has no control. If they walk into their office, say, I'm hearing voices, the doctor says, well, I think you have schizophrenia, and they want to leave, they can and never come back. And that's the problem, is they just don't subscribe to the treatment, and they don't want to hear that. So it's kind of a lost cause. But I mean, the idea. So
really, I mean, like the science communication that you're doing, though, Amal, is very generous of you to spend your time to actually respond to a number of these.
I will say one thing. There was one case, a legitimate case, where somebody was implanted against their will, and it was based on FUD, or fear, uncertainty, and doubt, or ignorance of what the chips are capable of. And it was actually a case where a sex worker was imprisoned by her pimp, right? And he went down to the vet office, got some pet chips, and just started putting them in the legs of his victims, and said, I can track you now, so don't try to leave. obviously not true, right? And so, but it kept through the fear of this, it kept them in the ignorance, it kept them from leaving.
It was
But,
true, or do you like? Was he? It was just a mind trick, like.
It was just a mind trick. It's not. He's just saying now I can track you, but you can't.
Needs a reader, yeah.
Yeah, absolutely cannot. I mean, if you get within 2 inches of the person, you could read it and go, there you are. But no, it was purely because they believed, they saw Mission Impossible. They know these chips exist and can be tracked. So that misinformation was used against them. But one person, I don't give a crap. I'm going to the hospital, and they went right to the hospital. Said, "Hey doc, I got a chip put in me by my whatever. I need to take it out." And they're like, "Yeah, whatever." Right? They thought she was mental, but then they did an X-ray, and they're like, "Oh wow, there is one." And they took it out, and they wrote a paper. And her story is like the only legitimate case where I could say somebody was actually implanted against their will. But yeah, out of hundreds and hundreds and hundreds, it's the only one.
Oh, very fascinating. And I'm surprised that there actually is a documented case where it happened.
Yeah.
So we're running short on time. I do want to ask you, all three of you, to speculate about the future. And kind of actually speaking of Mission Impossible, I mean, when that movie came out, it was what, 20 or 30 years in the future, right? So let's move not just in the next 5 to 10 years, let's move further into the future and speculate within your different fields, whether it's the ability to discern transcriptomics or even just all genetic data through software, tracking things through either genetic barcodes or these implanted barcodes. And before I turn the mic over, I want to front load the thank yous, because I always forget this part. Thank you so much to Josh Torrey and Lindsay Atkinson, who are running the show and making sure that all of the tech is running properly. We are very grateful for all of your help. And with that, I would be happy to let anyone go first to kind of take us into the future in terms of not only where you think the future is going, but opportunities for young, early entrepreneurs to move into.
I guess I could go first. I try to be very vague because in doubt, invariably you make a prediction about the future, it doesn't come true. And then people 50 years from now laugh at what an idiot you were. So I'll be very vague, but I think in general terms, it's very clear that we're seeing an acceleration toward integration of technology into our bodies. We're seeing scientific freedom to explore genetic alterations. I think that in terms of like where we want to go as a species, off planet, to the depths of the ocean, whatever, we might even see through these commoditizations of these types of technologies, divergence in our species, where we have people that are fundamentally at a genetic level branched off and doing their own thing. Spacefarers.
That is more than 20 to 30 years, I would imagine, but
well, I mean,
yes.
Exponential technology development is exponential technology development. You really can't tell if it's going to be 50 years or 300 years. I mean, it could be so fast. You look at the gaps between democratization of technology, like it took 50 plus years for everybody to get a refrigerator, but only really 10 years at most, maybe to go from the first commercial cell phones to like a lot of people having them, and now it's ubiquity. We don't have landlines anymore, and that's such a short amount of time, and it's only getting shorter as we progress in the future. So for me to say it's 50 years, that's dumb. It could be more, but it could be less for some of these things at least. I mean, there's genetically modified babies in China living their lives, like who's to say that it might be so long, especially if people go underground with a democratized, accessible technology. Who knows?
That's true. All right, I appreciate your vagueness. That's a very interesting perspective. Adam, would you like to make a prediction? And if 20 to 30 years seems far out, I mean, we appreciate even 5 to 10 years here, in terms of where you think different genetic manipulation tools, I guess we'll call them, or sorting tools.
So I'm going to do here exactly the opposite of what Amal did. I'm going to make a very specific prediction.
All right, we're here for it.
In 20 years' time, my ego is going to take a hit. So the prediction I'm going to make is that within the next 20 years, majority of patients in diseases such as cancer are actually going to use precision medicine. And what I mean by precision medicine is they will take an assay that is going to look at the molecular basis of the disease of the particular type of cancer or the particular type of disease that they have, and they will receive treatment which is tailored to that specific instance of the disease that they have. And more specifically, I think I believe that a single cell technique is going to become part of that precision medicine landscape. So when we look at single cell NAC, which is the technology that Biomage is built on top of, what this technology allows us to do is it allows us to look at individual cells in a sample, in a tissue sample. It could be a cancer sample, and it allows us to measure for each of those individual cells. It allows us to measure the full genetic picture of what's happening in that cell. And so we can measure a full transcriptome, or nearly a full transcriptome. And there are already techniques which are becoming commercially viable, where on top of being able to measure a full genomic information of every single cell in a sample, in a tissue sample, we can also get to know where they were positioned in the tissue. So this is called spatial transcriptomic. So on top of having those, as I mentioned earlier, 200 million data points, we're actually getting also information. On top of that, we're getting information on spatial coordinates of those different cells that we've worked with. So my prediction is I'm just really big on precision medicine. I'm a big believer in precision medicine actually happening within the sort of 20-year time scale. And I believe that single cell, some form of single cell technology is going to be a big part of this story.
Excellent answer. And I would easily go another, hour talking about some of those ways that it will be implemented, but unfortunately we are drawing toward the end. Thank you. And Vishal, could you please paint a picture of the future in terms of these genetic tags and how they might be implemented in ways well beyond what we can even do, what we are capable of doing today?
Perfect. So one, Adam, I totally agree with you on precision medicine. I think it's inevitable. And I've got sort of two predictions. One, company-wise, one of the things we're actually working on today, just signed a huge partnership last month with a company that will go public soon, is actually creating functionality for our tag. So not only do you have a tag, but it's now able to be antimicrobial, bacteriostatic, increase nutrients of the food. So we look at it as our tags as just a platform, and then we can add proteins as almost like biological apps. So part of just enhancing the food system and food safety. But sort of wrapped in that, the bigger prediction is on GMOs. So I think that the world is going to have to sort of change its stance on genetically modified organisms because of climate change. And I think it would shift it so anti-GMO sort of illogically because of things like Monsanto and sort of miscommunication around that. And now I think it's going to, and you're already seeing it with things like the Impossible Burger. Like, I think consumers are now starting to put kind of ESG and environmental friendliness and climate change above just a blanket, we don't like GMOs. And I think that's going to be a major tailwind for, our whole industry in our sector and specifically our business. But I think that, especially over the next, I think this will happen in the next 5 years or 10 years. But I think if, we're going to protect our crops and really like take on climate change head on, and obviously people's stances on climate change vary, but I won't get into that. But like, the way to do it is through GMOs. And I think that as a technology is just going to be, again, inevitable and something that everyone's going to have to embrace. And I think it's going to happen over the next decade.
Interesting. Obviously, we at IndieBio think the same because we are investing in that as part of the future. Wonderful. Well, I want to take a moment to thank all three of our panelists. This has been a really great discussion in terms of the many different ways that data and biotech can work together to create a better future. So thank you so much. And I should mention here that we're in 3 different time zones with our 3 different founders. So thank you for staying up working past the business hours there, Adam, over in Poland. Thank you for a kind of early morning for you, Amal, over on the West Coast, and Vishal, we're both in New York. So at least it's smack dab in the middle of our day. Thank you to all three of you.
Thanks a lot, Julie. Thanks.
Pleasure being here.
This was a lot of fun, and we will do it again. Stay tuned if you're interested in joining additional IndieBio New York events. There's a newsletter linked below. There's a little button that says our events newsletter. Go ahead and click that, subscribe, and join us next time. Thanks, everyone. Josh? Are we still live?